The last topic we need to talk about in this unit, genetics, is a little bit, just kind of a real brief introduction to DNA. Something in 10th grade probably you'll talk about a lot more, but we'll just sort of go over a few of the basics about what exactly DNA is, a little bit about kind of how it works. Okay, so DNA, like you guys told me as we answered our question of the day, DNA is something that has the instructions about how to build us, how our body is constructed, how our body works. All of that information is coded for in our DNA. Sometimes it's called the blueprint of life. What is a blueprint? So you've heard of a blueprint? Christian?
It's like the blueprint like helps you create your house and like shows you what designs that you
Yeah, exactly. A blueprint. You may not have seen an actual blueprint before. I don't know if they actually, yeah, they just do a story or something. But it basically has all, so a blueprint of a house has all the, it's not just a drawing of the house, but it's all the details about how thick to make the walls, what type of wood to use, where different rooms are going to be placed, where a door, which way the door is going to open. Basically, every single detail about that building is specified in the blueprints. Okay? And so DNA is kind of like that for our body. So you've seen blueprints before?
Fortnite.
What?
Why is this here?
Because he's building.
Yeah, when you build, your character there takes out their little blueprints when you're going to build walls and stuff, that's an example of blueprints. Oh, that's the Iron Man blueprints behind that guy. All right. So the word DNA, the three letters, stands for deoxyribonucleic acid. It's like basically DNA is just a very large, very long strand of a chemical called deoxyribonucleic acid. Go ahead and write that down. That's where DNA comes from. Deoxyribonucleic acid. No. And so where do we find our DNA? Where is it located? In our bodies.
Our nucleus.
It's in the nucleus of our cells, yes. So in our cells, which we talked about earlier in the year, we know there's a nucleus. Inside of that nucleus, there are these structures called chromosomes, which we inherit from our parents. Those chromosomes are made of DNA as one of their components. So that's where our DNA is stored. In the chromosomes, in the nucleus of our cells. And if you remember back when we looked at our cheek cells, we stained them, we looked at them in the microscope, that tiny little dot of the nucleus that you saw, inside of that, there's 46 chromosomes, and in those chromosomes has all of the information to build you. In that tiny little microscopic dot of the nucleus, all the information is there. All the cells in your body have the same DNA. A cell from your cheek has all the instructions to build your whole body. A cell in your liver has all of the instructions. Each cell has a complete cell. And on those chromosomes, we have this very, very long strand of DNA. And on the DNA, there are certain sections that we call genes that tell about certain aspects of our body, that tell about our eye color, our blood type, our hair color, how our liver produces insulin, and so forth. So we call those things genes. Christian?
Yeah. So there's many strands of DNA in our body, right? Not just one?
Right, every cell has them. So DNA was really not known until the 1950s or so. People understood that for a very long time that children inherit their traits from their parents. But for a long time they weren't sure how that happened. It was these two gentlemen who also had some help. Names are James Watson and Francis Crick. In the 1950s, they figured out the structure of a DNA molecule. They figured out its shape that we're familiar with today. Because we know if we see a molecule that looks like this, you probably already know that's DNA. That shape is called the double helix. It's kind of like a twisted up ladder or a spiral staircase, we sometimes say. And this molecule has the code for all of your genes. They're stored there in your DNA. The DNA molecule itself has these sides, if you imagine it's like a ladder, has these sides of the ladder. And then between it, the rungs of the ladder, the steps, are made of these things we call bases. And there's 4 different bases in our DNA. They're these chemicals, they're called adenine, thymine, guanine, and cytosine. We usually use a letter to abbreviate them. We use A, T, G, and C. And so the order of those letters are what makes you you. So the order of the letters in my DNA is different from the A's, T's, G's, and C's in your DNA. Many of them might be the same, but many of them will be different. And so the order of those bases is what gives us our traits. And the way that these bases match up, they match up in a certain way. Because DNA is a double-stranded molecule, we have our sides of our ladder, we have our bases in between. They always match up in a certain way. In DNA, adenine, A, always matches on the other side of the DNA molecule with a T. We say that those bases are complementary. And C, cytosine, always matches up on the other side with guanine. So to help you remember that, remember apples and trees, cars and garages. So if you know one of the DNA bases, you also know the one that's opposite on the opposite strand, because they match up in this way.
Oh, and those are customers for us.
Right. So as an example, here I have some DNA bases, A, T, A, T, C, A, etc. If this is one half of a DNA molecule, what letter is going to be here?
T.
Apples and trees.
Yeah, can you
What's
edit?
T going to go with? A. What's A going to go with? So the complementary bases for these 3 are T, A, T. How about what are the next 3 complementary bases? Alex? A, G, T. A's and T's together, C's and B's together. How about the next one, Julia? ACG. Zach, how about the last one? CAC. All right. So the bases match up with their complementary base in a DNA molecule. And the order of these letters are what give us our traits. Now, in that tiny little nucleus, there's actually a huge amount of information. If you were to take all the bases in DNA, it would take up about 3 gigabytes of space on a computer or on a phone. So if you have a phone and it's like 16 gigabytes and you run out of space, whatever. So 3 gigabytes would be our genetic code. And all of that fits in one nucleus of one cell. Tiny, tiny little thing. So the way that these genes work, a different set of letters is is a gene for a certain protein. So if a cat had CAT as its letters, maybe that is the code for purple hair. If it had a different set of letters, TAC, that would be a gene for yellow hair, and so forth. So the order of these bases are what give us our genes and our traits. It's kind of like we can put the letters of our alphabet in different order to make all the words that we have and make sentences and paragraphs, and you could continue that forever. The same is true of DNA. The order we put together these A's, T's, G's, and C's, that is what forms our genes. And what really genes do is they have the instructions, the codes to make a protein. They have the recipe to build a protein and tell when to build them. Our bodies are mostly protein. Enzymes are protein. Antibodies are protein. Protein is hugely important in our body. And so our genes tell how to make these proteins. What actually makes the proteins? Anyone remember way back to cells? Which cell organelle
Mitochondria.
makes protein?
Oh.
Give you a way to remember this.
Ribosome.
Yeah, the ribosomes. Ribs, protein, meat, ribosomes. And so what the ribosome is actually doing is it's reading the code about how to put together a protein and sort of following the instructions until the protein is complete. Now there are times when the DNA can get changed by accident.
Like the Teenage Mutant Ninja Turtles got mutated, right?
That's yeah, just like
That's
that.
fake.
Just like that. A mutation does not give one superpowers. It does not allow a small reptile to grow into a ninja. Sorry, yeah. But mutations change the DNA, and if you change the DNA, you change the traits. So sometimes this happens as an error. When DNA is being copied to make new cells, a mistake gets made. And I'll tell you about a couple examples of these mutations in a minute. But there's sort of three possible mutations that happen in cells when cells are divided. Sometimes a brand new base gets replaced for one that should have been there. So like a T gets replaced with an A, or a G gets replaced with a C. We call those substitution mutations. Sometimes a whole new letter gets just put in. We call that an insertion, because you're inserting a new base in the DNA. Sometimes a base gets removed and taken out. We call that a deletion, because you're deleting one of the bases from the DNA.
So did you make these up or are these real?
No, there's the name of that. That's what I call it. And so if you delete a base from the gene, or substitute an A for a T, or add a gene, well, you're changing the gene, and you'd also be changing the protein and changing the traits of that organism. There's some examples of diseases that are caused by a mutation. Here's one of them, sickle cell disease. So sickle cell disease is a disease in which there's a mutation in the gene that helps to create hemoglobin in our blood, the protein that carries oxygen. And if one of these letters gets switched, the hemoglobin is not produced properly, and the blood cells take on this strange shape, and they don't carry enough oxygen. And they clog up blood vessels, it leads to painful inflammation, it can lead to early death in a person that has this mutation. A friend of mine in high school, she had cystic fibrosis, which is a genetic mutation. It's a disease caused by genetic mutation, and it's somewhat common. And it's a mutation which causes the mucus in a person's lungs to be too thick. The protein gets changed, and it gets infected very easily. Person with cystic fibrosis is often congested. My friend in high school, she coughed a lot all the time. She had to go for regular treatments. She had to go on antibiotics a lot of times.
Do you want the vibrating vest?
Yep. Nowadays, I don't know that they did that when I was in high school. They sometimes have this vest they put on and it vibrates. And sometimes they'd go and somebody would, they'd get almost like a massage, or somebody would pound on their back and loosen up this mucus so they can get rid of it, so it didn't get infected. Okay. No, she passed away in her early 20s from cystic fibrosis. So then that was fairly common. That a person with cystic fibrosis often had a relatively short lifespan. Today it's improved quite a bit. So there are newer treatments. So they can't cure it right now, but there's better treatments that people live much longer with cystic fibrosis. Now, we'll talk about it in a minute, but new technology is getting us to the point where we may in the future be able to change that gene. So in my friend, one of these letters in her DNA was switched, which caused the protein that was made in her lungs to be switched, which caused all these symptoms. Well, if we could somehow go into her lungs and change that letter back to what it should be, then we could theoretically cure the disease. We can't quite do that yet, but we're getting closer and closer. So for you guys, as you get older, as you go to college and afterwards, this is going to be something that's going to be coming up, and something, if you go into sciences or biology or genetic technology, those sort of fields, that's going to become a growing field of finding ways to fix these genetic problems in people. And it could be a great way to help cure lots of diseases which are caused by things like these mutations. Alex?
Could they like have given her Mutations?
No, there's no real treatment. There's medications they would take to help, but there's no way of curing it. You can help treat some of the symptoms, but there's no cure. Christian?
So why there's not like a lot of people with it because of the people who
Yeah, it's not that common. You know, I don't know the likelihood of a person getting it. You know, it's probably less than 0.5% of people are born with that mutation. The gene has to be in both, it's recessive. The gene for that mutation has to be in both parents. And sometimes they do testing to see if parents have it. If it runs in their family, they may do testing before they have children to see if there's a likelihood they might pass it on. All right, so let's kind of review what we've learned about DNA right now. So DNA stands for, can anyone remember without looking the name? Nobody?
I know the one. What?
Hold on, can't give it a try.
Yeah.
Close. Deoxyribonucleic acid, DNA. Its shape. What is its shape?
String-like.
No. What's it look like?
Julia?
Double helix.
It's called a double helix. It looks like a spiral staircase, a twisted ladder. It was Watson and Crick who discovered that it's a double helix. Although, actually, interesting story, they had help from a picture taken by a woman, Rosalind Franklin, who never really received credit that she deserved in helping these male scientists discover the structure of DNA. They got the Nobel Prize. There's some debate as to whether they kind of stole her work. And so if you're ever interested in learning more about a woman scientist who contributed a great amount to our knowledge about DNA, it's an interesting story about Rosalind Franklin. This was in the 1950s. And at that time, there weren't a whole lot of women in advanced science at universities, and so often they were not treated the way that other male scientists work. And so it's an interesting story to learn about that. So the four bases are A's, T's, G's, and C's. A always gets together with T. G always with C. And finally, raise your hand if you could tell me what are the bases that should go along on the other side of this DNA molecule. Alexa? Yep. G, T, A, A, T, C. G's and C's together, A's and T's together. I'm gonna skip this.
Wait, how do you know that?
No.
No.
A's and
Can you go
T's.
back for 1 second?
A's and T's go together, G's and C's.
Oh, no,
together.
I'm stressed.
I gotta finish this. All right, last step. What I was just talking about, if we could go into my friend from high school and fix that gene in her lungs, that would be an example of something that we call biotechnology. Using new technology to actually alter genes. Now scientists have actually been, or not quite scientists, but even in agriculture, people have been altering genes for a long time, but not using technology, by doing what we call selective breeding. For example, where did dogs come from?
Wolves.
Wolves. Humans use selective breeding to breed all of our different breeds. By taking the smallest of dogs and allowing them to mate, then they would obviously probably have smaller dogs. And then if you take those litters, have the smallest ones mate, and continue that hundreds and hundreds of times, you end up with little tiny dogs, like a toy poodle. Or if you want dogs that can swim really well, you breed the best swimming dogs over and over again, and you end up with dogs that swim very well. If you want carrots that are really sweet, you take the sweetest carrots that you grew in your garden, you pollinate another type of sweet carrot with it, you grow the seeds, repeat many generations, you end up with sweeter carrots. All these colors of carrots came about because of this selective breeding. But nowadays, we have different ways of doing this. And one of them is called gene splicing. In gene splicing, we can basically take genes from one species and put them into another.
Let's go back.
Yep. So one example of a way this was done was scientists took the gene for human insulin. So insulin in our body helps control our blood sugar. It's a hormone. There's a code in all of our DNA that tells our body how to make insulin. Well, some people that are diabetic, their body doesn't make insulin. So they need to give themselves insulin injections or with a pump. Now back 40 years ago, they used to actually get that insulin from pigs. They would take slaughtered pigs, they'd take the pancreas from them, extract insulin, and that's what they would give people that were diabetic to give themselves injections. Some people had reactions, some people were allergic, it didn't work as well as human insulin. But since genetic splicing came about, what scientists did is they took from a person, they found the gene for how to make insulin, the instructions. They took it out of a cell, and they actually put it into a bacteria. And so now the bacteria has the instructions about how to make human insulin. And bacteria are easy to grow. They grow them in these huge vats, and then those bacteria are making human insulin, and scientists take that insulin, and that's what they give to humans now. It's actual human insulin. It's identical to the insulin you produce in your body, but they're having bacteria make it by doing this genetic splicing.
But isn't that what they do with trees? Like if the tree was like living or something, they cut it off and then stick it.
Like grafting, sort of like that, kind of combining two things. Now they've done this in lots of different ways. They've made glow-in-the-dark mice. They made goats that make spider silk in their milk by taking a gene from a spider and putting it into a goat. they can make, they can take genes for vaccines and put them into potatoes, and then instead of getting a shot for a vaccine, you just have to eat this potato. So there's all sorts of ways scientists have of using this genetic engineering and splicing. It's used a lot in agriculture as well.
How come they ended up being something from a bird to the fly?
Because you can't create like wings, it's smaller things. I guess we'll skip this for now. But we can also use DNA to help test family relationships. If you've ever heard of a paternity test, where it's unsure who the father of a child is, they can use a DNA
What
test
is that show?
to see who the father actually is. There's a show about that?
Yeah. There's a thing where it says,
Yeah, no, and then
Jim, yay.
All right.